All right, so we'll continue with the paper we started on Monday on MT5, and then we'll talk about another paper that you guys submitted about an interesting negative feedback loop triggered by A beta 42. But let's finish with MT5. So we left it here. We discussed how this paper is an interesting example of how there can be a massive rewiring of a gene regulatory network that controls the expression of, in this case, interleukin-1 beta, right? So we mentioned that we discussed that in wild-type animals, the expression of interleukin-1 beta is not controlled by these matrix metalloprotease MT5, hence the dash arrow there. So normally the expression of IL-1 beta is not controlled by MT5, but expression of IL-1 beta in response to IL-1 beta itself, this sort of self-inducing expression loop, is dependent on MT5. Hence, the arrow from IL-1 beta to itself goes through MT5. But then, if you have one of these mice that produces these APP that is prone to the amyloidogenic cleavage by gamma secretase, then two things happen. On the one hand, in that genetic background, MT5 now does control the expression of interleukin-1 beta in the absence of any pro-inflammatory signals. And if you do add pro-inflammatory signals to the culture, those can induce the expression of more interleukin-1 beta, but in an MT5 independent manner. So there's a complete rewiring of the network, and MT5 becomes disconnected from the interleukin-1 beta self-amplifying loop in these mice, these AD mice. So that's where we left it. And the next figure in the paper in a way revisits the same concept, but related to a different molecule, to interleukin-6. So we spent some time discussing two-way ANOVAs. I don't know how much you guys remember about them, but if we were to kind of predict or guess what a two-way ANOVA might show in this particular case, what do you think it would show? So let's break it down slowly. In a two-way ANOVA, fresh refresher, and maybe for those who weren't here on Monday, we analyze the effect of two different variables simultaneously, right? So in this case, these two variables would be the presence or absence of MT5, and the other variable would be being in a wild-type or a transgenic genetic background, right? So let me walk you through it so that it's not too awkward. Let's first talk about the MT5 effect. Do you think that MT5 does anything to interleukin-6 expression when you look at this data? Yeah. You think it does? Based on what? It goes up, right? And in this case, it goes down. So you're saying, yes, it does something to interleukin-6 expression. But if I were to ask you, what does it do? What would be your answer? Does it increase it or does it decrease it?
Does it depend?
It depends, right? And that's where the two-way ANOVAs become so useful. Because let's take a step back. What the two-way ANOVA does is it splits the data in such way that it pulls anything that refers to MT5 status with each other. So on the one hand, it will pull the data from the plus MT5 groups, and then it will pull the data from the no MT5 groups, right? If we do that, though, if we pull this data here with this data here, the average will be somewhere in the middle, right? And if we do something similar with this data here, I'm going to mark it with a little triangle, and this data here, the average will be somewhere in the middle. So when we do that, a two-way ANOVA might find that there is no effect of MT5. Because the two-way ANOVA will say, I'm going to put together all of the data that I have from samples that don't have MT5 in them and compare them to all the data that I have from groups that do have MT5 in them. And by doing that, it may turn out that there is not much difference. Okay? So my educated guess is that the MT5, or a two-way ANOVA would say that there is no significant effect of MT5. Then if we do wild type versus transgenic, what we might call the genotype or the genetic background effect, what do you think? Would there be an effect or not? If we go through the same exercise.
Can you repeat the question?
Yeah, so if we were to look at what the potential effect of the genetic background might be, meaning wild type versus transgenic, what do you think the two-way ANOVA might find?
Would it also be no significance?
I would agree, right? Because the split now would be in between the two halves of this graph. So it turns out that the middle point between the first two columns may be somewhere here, and the middle point between the second two columns may be somewhere here, right? So again, numerically, it may not be much different from that point of view, and I would suspect that there is no effect of the genetic background in controlling IL-6 expression. But like you guys intuitively mentioned, when we look at the interaction between these two factors, the MT5 interaction with genetic background or genotype, that's where essentially we're saying what the two-way ANOVA says, there's no effect of MT5 by itself, there is no effect of genetic background by itself, but when you put them together, there is a significant drop, right? This significant drop that you noticed before. So my educated guess is that yes, that interaction effect would be highly significant. Once again, indicating that the effect of MT5 is very much context dependent. What you guys saw intuitively with your eyes that, well, when I take out MT5, it seems like IL-6 expression goes up in a wild-type animal, but if I take out MT5 in a transgenic animal, it goes down. So, Lara, you said it kind of depends, right? We call that context-dependent effect. Okay? And it's, once again, a strong indication there's been some massive rewiring of the network controlling, in this case, the expression of IL-6. Yeah, so these are cells that were extracted from mice that are either wild type, first two groups, or transgenic. And when we say transgenic, what they do is they carry a mutant version of APP that is cleaved in a way that generates A beta that is toxic, the 40 and the 42 versions, right? So these are experimental models. These are transgenic mice that were made on purpose so that they would generate more of these A beta that is toxic. So it's a mouse in which you're forcing, in a way, the development of Alzheimer's. Okay, so what I have here down here is simply a schematic representation of what you guys saw intuitively, that MT5 seems to be inhibiting IL-6 in the wild type background. Again, we're going to do a split here in the middle between wild type and transgenic, but it seems to be inducing IL-6 expression in the transgenic background. And we say that because in the wild type background, when we take out MT5, there's a noticeable increase in the expression of IL-6, but in the transgenic, when we take it out, there is a noticeable decrease in IL-6 expression. So MT5 flips from inhibitor to activator of IL-6 expression in the transgenic cells. And I can think of a more clear, more explicit example of a rewiring of a network when you're turning a switch between an activator to a repressor or vice versa, right? Okay, so everything that we've discussed so far was at the mRNA level, and we've sort of made the conceptual leap to assume that it may be reflective of things that go on at the transcriptional level, right? That these things may be reflecting either transcriptional or post-transcriptional regulation of gene expression. So all of these regulatory connections that we mentioned about interleukin-1 and interleukin-6, both in the absence or presence of MT5 and in a wild type or a transgenic mouse, they all refer to mRNA levels. What about the protein level? So for reference, I'm bringing back the data about interleukin-1 mRNA levels, but then they actually looked using an ELISA assay. They actually looked at the levels of IL-1 beta, the interleukin-1 beta protein in the supernatant of these neuronal cultures from the different genetic backgrounds, right? From mice of different genotypes. And what we observe is a profile that is kind of different from what you see at the mRNA level. And I would like to particularly bring your attention to what happens in the MT5 knockout mice that are in an otherwise wild-type background, right? And the reason I want to bring your attention to that is because at the mRNA level, it seems like there is no difference in interleukin-1 mRNA between the mice that are not exposed to IL-1 and the mice that are exposed to IL-1. It's almost like these. In these cells, it makes no difference if the cells are surrounded by IL-1, but then when you look at the protein, there is this significant increase in this jump in the amount of IL-1 present in the supernate. So my question to you, and there is no single correct answer, or there is not one correct answer, mechanistically, how would you explain that? That the levels of mRNA are comparable between the two conditions, no interleukin-1 in the culture media or interleukin-1 added to the cultivation media, and yet when you look at the protein level, there's this massive difference. What could be going on in these cells to explain this difference? I'd like to hear other voices. Ale? Inhibitor. So you say that MT5 must have been some sort of inhibitor of, you mentioned translation. Mm-hmm. Now we take it out and the mRNA is being translated. More protein degradation in which case? In the control, right? So more protein degradation. So in cells that lack MT5, for some reason in the controls there is more protein degradation than in. Of course, and I mentioned at the beginning there's more than one possibility. Lara, you had your, yeah? Any other ideas? At all. And just if you hold that thought for a few seconds, we're going to address that question. Yeah. But you notice incorrectly. Yeah. So stability, yeah. Emily mentioned the idea that maybe in the control cells the protein was being degraded more, that this interleukin-1 was being degraded more. Yeah, I would call differences in the rates of degradation a form of stability. Yeah. And once again, all of these things are symmetrical, right? So one could say that the interleukin-1 protein is less stable in the non-inflamed neurons than in the inflamed ones, or that addition of interleukin-1 to these MT5 negative cells stabilizes the protein. So it can go either way, mechanistically speaking, right? Cool. So yeah, absolutely. It could be differences in either of those things. But as Arshia was pointing out, the one thing that may catch our eye about these two graphs is that when you look at the gray bars, the profile of the gray bars, that went too fast. The profile of the gray bars is very similar between the two graphs. It's the black bars that are very different, that are showing differences in mRNA levels within the non-interleukin-1 exposed cells and the protein that is flat across the board. So, Arshia, since you noticed it and you brought it up, what do you think might be going on? And okay, so something that accelerates protein making, but is clearly not present in the black bars, right? What could be that thing that accelerates protein making that is absent in the black bars? Exactly, right? So the idea here is, what you're saying is maybe there is something that accelerates or that, I would take it a step further, I would say that allows for protein production, that allows for translation, and that might be the addition of interleukin-1, right? So a very interesting example of how the various genetic backgrounds contribute to differences in mRNA levels of this gene, but the absence of interleukin-1, or more likely some sort of inflammatory state in these cells, completely negates the translation of these mRNAs for interleukin-1, no matter how much there is. Right? So notice how at the mRNA level there are all of these differences. In this graph it's a little less, it's a little harder to appreciate, but in the previous graph at the difference scale, we had seen how there were some significant differences across these groups, right? Which led us to say that there is a rewiring of the network and so forth. So it's a little less appreciable at this scale, given the presence of the other experimental groups. But there were some differences in mRNA levels across these samples, and yet at the protein level, everything flattened out. And once again, it may be simply an indication that these mRNAs may be present in these cells no matter at what level, but there is something above them that says, no, you're not going to translate these mRNAs. And that repression of translation is lifted when you incubate the cells with interleukin-1. And what we know from previous conversations is that when you incubate cells with interleukin-1, what you're doing is you're inducing an inflammatory response in these cells. When you say the MT5 knockout, I'm assuming you're referring to this guy here?
Yeah, it's just a visual, right?
And you're saying that it makes more protein, but makes more protein than this guy here? Is that what you're saying, or?
Yeah, so it's bigger than the mRNA.
Oh, yeah, bigger than the mRNA. But we shouldn't ever compare bars between graphs, because the scales are completely different, the variables and everything. Yeah. Relative heights, sure, we can do that all day. So one thing that you could say is, well, hold on a second, MT5 knockout cells have less mRNA than the MT5 knockout cells in a transgenic mouse, yet they have more protein. That we can do. But comparing it across graphs, that would be a no-no. Relatively, it could be. Now, one thing that I should say, though, is that notice what I just said, and your intuition hangs from determining that these two are significantly different from one another. So when I make the claim, look, MT5 knockout cells produce less mRNA than the MT5 knockout cells in a transgenic mouse, that's a strong statement that I may or may not be able to back up with statistics, right? So the fact that they didn't draw a bracket signaling or indicating a statistically significant difference between these two, they never said, oh, by the way, we compare these two and they are significantly different. Since they didn't do that, there is always the chance that those two may look different to you, but those differences are not statistically significant. That's why we do statistical analysis, to not have to trust our eyes or our judgment and let math speak for us. right? So the same, of course, goes to saying at the protein level, the MT5 knockout cells are producing more protein than the MT5 knockout cells in a transgenic background. In that statement, I'm implying that I analyzed these things statistically and that I found statistically significant differences, but maybe not, right? So even those within variable relative comparisons, you need to back up with statistical analysis. Oh, so what are you thinking? So intuitively, you're thinking, okay, so for IL-6, we said there was a switch, right? And now, in a way, we're saying there's a switch, too, of sorts. Is that the connection that you're trying to make?
I mean, I don't see a switch necessarily here.
Arshia saw one, right? And we spent some time saying, well, but we don't know if it's real or not because we didn't run the proper statistics. But maybe with a larger sample size and more careful analysis, who knows? There could be some sort of switch.
I think I'm thinking of the IL-6, but
Oh.
that's not here.
Not here. So then you're in a way answering your own question. In that these two things, what happens to IL-6 and what happens to IL-1 at both the mRNA and the protein level may be simply disconnected or unrelated.
Okay.
But I love the thinking, the whole, I'm making all of these observations, could there be a thread here, a narrative connecting the things together? But you answer your own question. Well, with interleukin-6, there was a switch between the wild type and the transgenic. I don't see a switch necessarily here between the wild type and the transgenic. So what that might say is whatever is going on with IL-6 can't easily explain what's going on with IL-1 or vice versa.
Okay.
Cool. Other questions or comments? Need for clarification? All right. So, when they saw so we're going to revisit something that we just discussed, but let me bring us all to the same page again. So when they saw the profiles of protein in the culture media of these cells, it immediately begged the question, why the differences in secreted interleukin-1 levels across samples? And one possibility we already discussed, and that is that if we ignore what happens in cells that are not in the presence of interleukin-1, so when we ignore what happens to the cells in the absence of any pro-inflammatory signal, because in the absence of any pro-inflammatory signal, there may be some sort of negation of translation of any interleukin-1 mRNAs, and we focus only on what happens with interleukin-1 protein in the cells that were exposed to an inflammatory signal, then there is a pretty good correlate between the mRNA profile and the protein profile, right? And the reason I say this is because if it's just as simple, more mRNA, more secreted interleukin-1, less mRNA, less secreted interleukin-1, at least we don't have to go crazy about thinking what may be different from a central dogma of molecular biology perspective, differences in transcription or translation. It's very straightforward. More mRNA, more protein. That's it. Right? Do you guys see it? So when we consider both culture conditions without interleukin-1 and with interleukin-1, it was really confusing because the mRNA in the cells not exposed to interleukin-1 was all over the place, yet protein was flat across the board. So what's happening there? But then if we ignore that for a second and we take that easy way out of, well, maybe in cells that don't see an inflammatory signal. They just don't translate interleukin-1 mRNAs. Let's focus on what happens when these cells are under an inflammatory state. Then there is a pretty good correlation between mRNA levels and protein. So that seems relatively straightforward, right? But then being scientists, they decided to look at not the secreted interleukin-1 protein present in the culture media, but the total interleukin-1 protein that they can detect in cell lysates, right? And that was a problem because what used to be differences in protein expression that closely matched differences in mRNA levels became a nearly complete absence of differences across conditions. So the first 3 experimental groups show no differences among them, even though the levels of secreted interleukin-1 are all over the place. And then, ironically, there is that sort of complication in the 4th group, the transgenic mutant for MT5, that shows statistically lower levels of total protein production, let alone what gets secreted to the outside. They had less protein overall, right? So, but just give me 1 second. I'm gonna tie up this thought, and then I'll listen to you. So back to the drawing board in a way, right? Because for a second there, we thought it's just that simple. More mRNA, more protein, less mRNA, less protein, we can move on. Not that simple again, because when we look at the total protein production, it turns out that for most experimental groups, they all produce the same amount of protein, but it seems like the difference is in how much they put out. Yes, Sergio. So the wild type and the MT5 knockout are different mice, right? And these bars come from data obtained from cells that were purified from these mice and placed in culture, okay? So when you say, are they from the same thing? No. So they come from different mice lines or mouse lines. What is the difference between a wild type mouse and the MT5 knockout? Presumably the only difference is that the MT5 knockout doesn't have the MT5 gene. That's it. We know a little better. We know that with experimental animal strains, be it mice, flies, worms, plants, anytime that you keep a given genetic stock in a lab for a while, they're going to be accumulating additional mutations. So I bet that there are more mutation or more genetic differences between these two than only MT5, the presence or absence of MT5. But on paper, the only difference between those two is just one mouse has MT5, the other one doesn't. Yeah? The controls too. So when we talk about controls versus IL-1 exposed, the black bars versus the gray bars, the difference there is just that you had your culture of neurons that were obtained from these animals, and either you dump some IL-1 into those culture media or not. But genetically speaking, in that case, yes, those cells were identical to one another. Oh, so it's the way that these mice were made is, on the one hand, you have your transgenic line, right? That if you did things correctly will be homozygote for that transgene. So you will have 2 copies of this variant of APP. And then you have your MT5 knockout line, meaning that mouse is homozygote for the MT5 deletion. So neither of its chromosomes that would normally carry the MT5 gene do. Okay, so far so good? So you have those 2 mice. Those were generated most likely independently, right? And you can obtain those 2 mice. And in your lab, what you do is you cross them to each other. So you have your MT5 minus over an MT5 minus. So this is a mouse that is homozygote for the deletion, and you cross it to a transgenic homozygote. Okay? I don't know where this transgene is in the mouse genome. I don't know where the MT5 gene is in the mouse genome. I don't even know how many chromosomes mice have. But I bet they're not on the same chromosome. For simplicity of the argument, they're not in the same chromosome. So when you cross these things, what you're going to end up with is a transheterozygote, meaning you will have a mouse that is progeny that is MT5 minus because they inherited the lack of MT5 from one of the parents, but then they're going to be wild type for MT5 from the chromosome they got from the other parent, right? And then they'll be transgenic over plus. So far, so good? Okay. I know you took my 360 and we went over this, but for those of you who didn't, when you're working with mice in an experimental genetics lab, one of the things that people do all the time is something called genotyping. And what that is you use molecular genetic techniques like, say, simple PCR to find out if a given transgene or a given gene is present in your mouse or not. The reason I say that is because once you have these mice here, you can cross them to one another. All of the progeny, if your original MT5 knockout was, as any knockout line, homozygote for the deletion, and your transgenic is homozygous as well, the entire progeny of that original cross will be transheterozygote, right? Or double heterozygote. When you start crossing brothers and sisters from this litter of pups, obviously you wait until they reach sexual maturity and then you let them mate one another. Then, from a Mendelian perspective, you're going to have all kinds of combinations. You're going to have some MT5s meeting up with other MT5 minuses. You're going to have some transgenics meeting up with some other transgenics. But then you're going to have some mice in which everything is wild type and some mice that reproduce these double heterozygote genotype. How can you tell them apart? Well, that's where genotyping comes in. You take a little sample from them where you can extract enough DNA to do some PCRs. And with PCR, you can quickly ask the question, is the MT5 gene there or not? If you do that by PCR, if the PCR works, it means that the MT5 gene was there, and therefore you don't have an MT5 deletion. If you see no PCR band in those mice that you're genotyping, it means that those mice are mutant for MT5. They're knockouts. And the same goes for the transgene. With transgene gets a little more complicated because you need to design your primer such that it allows you to distinguish the presence versus absence of the transgene, but that's totally doable, right? It's really like not a big deal, and that's what you do through PCR. You establish which ones were your double, in this case, double mutants, right? So homozygote transgenic and MT5 knockout. I say all of this, it took me maybe somewhere between 45 seconds and a minute and a half. In real life, it's something that would take somewhere between 6 months and 9 months, but it's doable. A PCR takes 3 hours tops. What with the 3 to 6 months is the whole, I'm gonna cross these mice and hope that some of them are double mutant, and then you do it and you don't get any, so then you do the cross again, and so forth.
That's why they work with flies
Or yeah. And so there are some model organisms that will give you an answer within a day. So flies will still give you answers within a couple of weeks or months, right? If you work with yeast, you have the answer within 48 hours. But so why bother with mice then? Well, because we're a little closer to mice anatomically and physiologically than yeast. Cell biology. So I always half jokingly, apologies everybody for the deviation, but these are important questions. I always say the same when I introduce the research in our lab to people. I say, if you look at a human being and a fly, can you tell any differences between them? I bet you can, right? I bet you can. Cool. If I show you a fly heart and a human heart, do you think that you can tell them apart? Yeah, if you're curious, the human heart looks like, the four. Now I got myself into this trouble. I'm gonna draw a human heart, right? So the 4 chambers and the aorta going here and whatever, right? A fly heart, fly heart is simply an engrossment of central, of a muscle that simply bubbles up and pumps. That's all it does. No chambers. So yes, you can tell them apart. If I show you pictures of fly cardiomyocytes and human cardiomyocytes, you're in trouble. Because now you can't tell them apart. They both look like striated muscle, and the nucleus to a side, and all those T microtubules. You can't tell them apart. And if I show you the inside of a fly cardiomyocyte and a human cardiomyocyte, even worse. Because now you're looking at proteins that are almost 100% shared between the two species. Right? So, yeast, same argument. A yeast and a human, very different. Yeast doesn't even have organs. But when you go down and down and down and down, at some point you hit that the structure, the subcellular structure of a yeast cell and the subcellular structure of most human cells is the same. A nucleus, a plasmic reticulum, Golgi apparatus, mitochondria, blah, blah, blah. Yeah? So yeast is super useful for those cellular biology questions. Flies, super useful for certain developmental biology questions and certainly genetic regulation questions. Mouse is when you get into the physiology and organismal biology questions, because their organisms are much similar to ours than a fly's. So these authors were a little puzzled by the fact that there were no differences in total interleukin-1 production, yet there were differences in secreted interleukin-1 levels. So they say, and I quote verbatim here, these results raise the possibility that interleukin-1 beta may be more efficiently eliminated in the microenvironment of those cells lacking MT5. And they actually propose a hypothesis. They say maybe MMP2 and MMP9, which are closely related to MMP24, the acronym, the alias for MT5. Maybe in the absence of MT5, these two MMPs get upregulated, and that's why interleukin-1 is being degraded more on the outside of cells lacking MT5. So they proposed that hypothesis. So what they did is we asked whether generally proteolytic activities located in the extracellular environment could explain a differential degradation of interleukin-1 beta. To this end, we incubated recombinant interleukin-1 beta for 24 hours in cell-free conditioned media from cell supernatants. So what they had is the different cell cultures from different genetic backgrounds. They collected the conditioned media that presumably would have more or fewer proteases in them, and then they dumped into those conditioned media recombinant interleukin-1 beta. So they produce interleukin-1 beta somewhere else, and then they mix it with this conditioned media. And the question that they were going to be asking 24 hours later is, how much interleukin-1 beta do I have left? Right? Because if there were different amounts of proteases that could chew up interleukin-1 beta in the conditioned media of the MT5 missing cells, then you would expect that you would have less interleukin-1 beta surviving. But when they did that, interleukin-1 beta content remained unchanged over a 24-hour period. So the difference between MT5 knockout cells and is not proteases on the outside. They said, well, maybe it's differential degradation on the inside. It could go either way.
Yeah.
Well, good news for you, it went no way anyway, because they saw no difference. So when they repeat the exact same experiment, but instead of exposing these recombinant interleukin-1 beta to the outside of the cells, they exposed it to whatever was inside of these cells, to the lysates. that interleukin-1 beta did just fine. Nothing was degrading it. Okay? So no differences across the groups. So it's nothing on the outside, nothing on the inside. What could it be then? If it's nothing floating around outside, nothing floating around inside, what could it be? Cell membrane. Cell membrane. Jessica? So, export of what? Of IL-1 beta, that's right. So that's a whole different scenario. Love it. So maybe it has nothing to do with how much it's being destroyed outside, inside, or at the cell membrane. Maybe is, yes, the cells are producing the same amount, they're just pumping out less when they lack MT5, right? Excellent idea. My suspicion is that they never considered that possibility, because if you're exporting less in the MT5 minus cells, you would probably expect to see more inside of them. But when they looked inside of them, at least when it comes to the wild-type background, it was actually the same amount. And when they looked in the transgenic, the double mutant background, it was less, not more. Right? So your idea is conceptually correct. I think that they may have strayed away from it by looking at this data and saying, well, it doesn't pair up with what we're seeing. There's no IL-1 beta that is getting stuck in these cells. So they're sort of hang up on this idea that the cells are producing the same amount, but the reason why MT5 minus cells, you see less interleukin-1 beta on the outside, they thought it may be proteases that live outside. No, it wasn't. It may be proteases that live inside of the MT5 minus cells and are chewing it up before we can export, the cells could export anything, but it wasn't. So now we have this idea that maybe it could be proteases that live at the membrane. And that's something that they didn't test. That's something they didn't test, but it's certainly a good idea. Questions or comments, anyone? All right. So finally, when it comes to the interleukin-1 and the whole neuroinflammatory angle, if you guys recall from Monday, this group said MT5 could be linked to all three pillars of Alzheimer's disease progression, right? It could be linked to neuroinflammation, it could be linked to amyloidogenic APP processing, so production of A beta plaques, and it could be linked to hyperexcitability of neurons. And in the paper, they decided to test all three. Everything we've discussed so far is about neuroinflammation. I'm not showing the data because it was even more confusing than with IL-1 beta stuff. So they have one figure and they're like, we don't know what's going on. About the excitability of neurons, they did measure and they found differences, but it's all over the place. And then finally, they said, let's look at APP processing. And to make a long story short, they simply went for same genetic backgrounds as usual, and they measured the amount of A beta 40 or A beta 42, the toxic forms of A beta, in the culture media of these cells. And luckily for us, this time there were no differences based on genetic background. What happened across the board is when the cells didn't have MT5, either in a wild-type background or in a transgenic background, there was less of these toxic A beta forms being secreted out, right? So there was less of A beta 40 and A beta 42 outside. So in terms of new connections for a little network, we can say with some degree of confidence that MT5 induces somehow or leads to production of the toxic A beta oligomers. Why? Because when you take it out, in either genetic background, and when you're looking at either toxic isoform of A beta, you see a significant reduction. So that means that MT5 normally promotes the production of these things. When you take it out, you'll see less. And then based on all the previous data, somehow MT5 might be positively connected to the production of IL-1 beta, particularly when it comes to this business of sending more to the outside. So when we take out MT5, now the cells are sending less IL-1 beta to the outside, indicating that somehow MT5 may be promoting that effect. Though, I should say that this huge question mark here, because there is all of those confounding effects of whether we're talking MT5 in a wild-type background or in a transgenic background, we're talking mRNA or protein, so it gets a little complicated that way. And since there are some experiments missing as to what the connection might be, whether it's proteases on the membrane or differences in whatever else, I don't think it's prudent to add that particular connection. But that's what the data might indicate, that there is some sort of positive connection between MT5 and interleukin-1 beta. Now, a question for you guys. Over and over, we've seen that when you incubate brains with interleukin-1 beta, this is not the first time that we've seen people adding interleukin-1 beta to their biological systems. There is an increase in inflammation, right? And we've seen before that inflammation leads to higher production of A beta toxic forms, right? We even have it in our little network that the TREM2, the inflammatory receptor, induces the production of A beta and so forth. Here, when we look at the production of the toxic A betas in response to interleukin-1 beta incubation, there's no change. Look at how across the board, across treatments, across molecules, there's no difference between these guys. They're always the same. So it's like IL-1 beta is not doing anything to A beta 40 or 42 secretion. My question to you is, how come? Why didn't interleukin-1 lead to higher expression / secretion of of toxic A beta peptides? Ale?
Maybe it takes some time.
Take some time. All of this is Petri dish, yeah.
And maybe it's not like directly the cause to the IL-1 beta. If there's other companies that still need to occur between those steps, that might be inhibited.
So we
or however.
I like it. So you're saying there's these steps. Adding interleukin-1 and production of the A beta. One possibility is that we didn't let enough time go by. Another possibility is that we need intermediate steps that are not there. What could be not there in this experiment? You have the interleukin-1 beta that you added to the cells, and you're asking whether the cells are producing more interleukin-1 beta. So what could be missing? Kind of relates to the idea that all this happens in a Petri dish. So what's missing in a Petri dish? And what's in here?
The actual tissue that it's occurring.
Okay.
I would say the information encapsulating.
So here's what we have. What's missing is the brain. The brain can go into a panic signal. It can go into some sort of response to that inflammatory signal. What inside of the brain responds to inflammatory signals, Norman? The neurons? If you go the neurons and I go the neurons, what's the other one? The microglia, the astrocytes, right? So those glial cells that are the ones that normally mediate neuroinflammation. So what this is telling us is that in all of those previous studies in which anything that would send a stress signal, an inflammatory signal inside of the brain that would trickle down to maybe even neurons producing more A beta toxic peptides or tau neurofibrillary tangles, that may have been mediated by these other cell types that talk back to the neurons and say, hey, you should produce more A beta or something like that, right? And that's missing here. So in a way, this relates back to an idea that was thrown around a couple weeks ago, that when we make these connections between genes in these networks with information that we obtain from complex systems with various cell types, we need to be extremely careful, because connections, genetic connections that exist between genes within a given cell type may not exist in others. And then there may be genetic connections that happen at the organismal level, but that are explained by communication between cell types, right? So communication of the type that if a microglia gets activated by interleukin-1 beta, then it will send some sort of signal to the neurons that says, make more A beta plaques and more neurofibrillary tangles, okay? And that's missing in a Petri dish that has nothing but neurons. Okay? All right, now I don't know how long we'll get ahead with the second paper, but I think we have enough time because this paper is super straightforward. So the second paper that I chose for this week, Alzheimer's disease linked A beta 42. If you guys remember, 42 is one of the most toxic forms of A beta. Exerts product feedback inhibition on gamma secretase, impairing downstream cell signaling. So let's see what this paper is about. In the introduction, they say that gamma secretases process other proteins in addition to APP. We heard about gamma secretase in this class because it's the one that cleaves APP in a manner that generates toxic A beta peptides. But it turns out that gamma secretase is in cells, present in cells to process other things as well, not only APP. Previous work showed that treatments with gamma secretase inhibitors, or GSIs, worsen AD in patients. And I didn't check those references, but I can almost imagine what happened here. When people saw that gamma secretases produce toxic A beta, they said, let's inhibit gamma secretase in AD patients, for instance, or in AD mice, because things should get better, right? They got worse. So AD patients or mice treated with GSIs, that worsened the problem, okay? And the other thing that they mentioned in the introduction is that BASE, one of the enzymes that cleaves APP, first cleaves APP to generate APP, that's missing there, so I'm going to add it here. So this should be APPC99. So, or CTF, they go by the same. So this first cleavage here by the beta secretase or base generates a fragment called APPCTF or C99. And that's the one that gets further cleaved by gamma secretase, somewhere here in the middle, to generate the A beta, anywhere between A beta 37 to A beta 43. And what those numbers indicate is the number of amino acids that end up in those fragments. And why that is because gamma secretase can cleave the APPCTF in various places, right? So which one you end up with depends on where gamma secretase cleaved. In general, longer peptides are more prone to aggregation and poor clearance. And by clearance, I mean those pumps that would take toxic A beta peptides and pump them out from the brain and the cerebral spinal fluid, put them in blood and let the liver deal with it. And what the authors hypothesize in this paper is that A beta 42, the toxic form of A beta, can inhibit gamma secretase and contribute to AD progression. If you ask me, I think I noticed you're like, what? I feel the same way. This was something like pulled out of a hat because they're telling us about all these things. I don't see the logical connection here. But they're like, and by the way, we hypothesized that, so I don't know if it's one of these cases in which they had some preliminary evidence that they didn't want to share. I don't know. But they're like, this is what we know about gamma secretases. This is what we know about APP cleavage. By the way, we hypothesized that AB42 can inhibit the enzyme. Okay. So one question that I throw out there right off the bat, in the formulation of their hypothesis, the authors say AB42 will inhibit gamma secretase, and that will contribute positively to AD progression. My question to you is, if we inhibit gamma secretase, and presumably we're going to be reducing the amount of new toxic A beta being produced, how can that worsen AD progression? To exit the cell. So you're saying it produces it, but it also helps it to exit.
Not directly, but it has something to do.
Okay. Okay. So like gamma secretase directly produces AB or A beta, but indirectly may contribute to its clearance.
Maybe like, well, it makes less A beta. It also like inhibits other things like.
And that's in the title of the paper, right? Exactly. These guys think, yeah, it's so impairing downstream cell signaling. What they imply, and they will show us later, is that it's not about what happens to A beta, it's what happens to cells when you inhibit gamma secretase, right? So sure, you're making less A beta, but you're also making a lot less of who knows what that is making these cells sick. And at the end of the day, AD progression is about neurons that are getting sicker and sicker and sicker. Right? Cool. So, first things first, can AB42 inhibit gamma secretase? What they do is they come up with an in vitro assay. They take APC99, the substrate, and they can detect that by Western blot. They have it in a test tube. They incubate that APC99 substrate with human A beta 42 at various concentrations. And as the concentration of A beta 42 goes up, there is less and less and less APC99 present in those tubes. So there's this negative correlation between the amount of A beta 42 present in the tubes and the amount of APC99 that is produced. I realized there that I call APC99 and CTF synonyms. They're not. APC99 is the other. No, actually, that is the substrate. I apologize. That is the substrate. And what they're measuring here is how much of that substrate is released. There you go. So with less of that substrate being released, that means that A beta 42 was inhibiting the reaction. There you go. So they test that in vitro, then they do a similar assay in vitro, but this time making different kinds of gamma secretases. So gamma secretase, we say gamma secretase, but gamma secretase is actually the name of an enzymatic complex that is made of presenilins, either one or two, and then these other proteins. So they tried all kinds of combinations to make all kinds of different gamma secretases, and therefore asked the question, will A beta 42 inhibit them all? And to make a long story short, yes, it does. Anytime that they added A beta 42, that means here and here and here and here. Every single time that they added AB42, no matter what kind of gamma secretase they were adding it to, they inhibited its activity. And in this particular experiment, they have the GSI as a positive control. They wanted to make sure that they could see inhibition of gamma secretase in this in vitro assay, and they're adding this GSI, and they can see the inhibition. So that's a positive control. Why do you think they wanted to work with purified gamma secretase, like in this experiment, and not with whole cells?
So the
But so what do you mean? The gamma secretase would be in the cells, in whole cells. So why wouldn't they be able to see the inhibitory effect of A beta 42 on gamma secretase if it is in a cell? So in Jacob's case, there may have been things that were already cleaving stuff. In your case, maybe other things cleaving stuff, right? And by the way, you were right at the beginning. When you work with, I was just messing with you. When you work with cells, you never know. When you add A beta 42 to the cells and you see that there is less gamma secretase activity, you never know. It may be that A beta 42 is regulating the transcription of gamma secretase proteins or translation of gamma secretase, and it has nothing to do with inhibiting the complex itself. So whenever you want to make a claim about molecule A inhibits molecule B, the best approach that you can often take is isolate both, purify both, and test them in vitro. Then you'll get a whole bunch of people saying, "Whoa, but that's in vitro. How do we know if it happens in vivo?" Then we'll cross that bridge when we get there. And these guys did cross the bridge when they got there. They tried these same experiments next, but in cells. So what they did is a little fancy, but essentially what they did is they created cells that have or that produce not in this experiment, in the next. So they took cells, they incubated the cells with various levels of A beta 42 to different concentrations, or of an unrelated peptide that can also be produced by gamma secretase that is called P3, right? So sometimes, depending on what substrate you give gamma secretase, the peptide that it produces, and when you give it the APP-CTF, it will produce AB42, but if you give it something else, it will produce this P3 molecule. So they try both, and long story short is when they measure the ratio of APP-CTF to full-length APP, what they saw is that in the presence of AB42, there was a significant increase in that ratio. What that means is that to equal levels of full length, there was an increase of CTF. That to them is an indication that that CTF is being processed less. If CTF is not being properly processed by gamma secretase, it will start piling up, and that shows up as an increase in the ratio of CTF to full length. Does that make sense? If they had measured CTF by itself and saw an increase in CTF by itself, there's always a devil's advocate that says, well, but when you give A beta 42 to cells, they may start producing more APP. So you're going to see more CTF because there's more APP. And they're like, okay, let's do a ratio to full length. So if I see an increase in the ratio of CTF to full length, it means that there is this accumulation of CTF, which I interpret to mean that that CTF is not being processed by gamma secretase. That's on the substrate level. They also looked at the products label, so they actually looked at the amount of A beta 42 that was being produced. Here's where the experiment got tricky. In order to distinguish between the AB42 that is being produced by the cells and the AB42 and not confuse it with the AB42 that they are adding to the cultures, they needed to make sure that they can distinguish between the two, and they came up with a transgenic cell line that expresses an AB42 that is tagged with an enzyme that can produce light when you give the right substrates. So in this case, they're measuring light, and what they see is controls and cells treated with this non-toxic P3 peptide produce a lot of light, right? So very high levels of luminescence, but when they incubated the cells with A beta 42, the amount of light produced was less, right? Significantly less, indicating that there is less production of new A beta 42 when the cells are incubated with A beta 42. Hence, this idea that this is a product negative feedback loop. The more AB42 there is, the more gamma secretase will be inhibited, the less that I will produce moving forward. Right? We're not going to make it, so I'm trying to see what's the best way. Maybe we can stop here. I'm going to skip a bunch of things, and I will simply say this. Remember, Jacob, when you said if you're making less A beta 42, that's good, but maybe you're stopping gamma secretase from doing things that are important. It turns out that they looked into other signaling pathways, and one pathway that was affected was the processing of a receptor called P75. And it turns out that when this receptor is not being properly processed, it can send a cell death signal to the neurons. But only if those cells are not activating another receptor called TRK. So to make a long story short, when they incubated the cells with A beta 42, nothing happened to the survival of those cells. But if they incubated the cells with A beta 42 and an inhibitor of TRK, now those cells are dying. These bars show cell death, right? So what those experiments showed is that if you add either A beta 42 or GSI, and you inhibit TRK in these cells, now they're dying. And in their opinion, that's an explanation for why inhibiting gamma secretase, which would normally be a good thing because you get less A beta toxic, can also be a worse thing because you're taking away survival signals. And in culture, in the absence of any other damage or stress, the cells do fine, but inside of a brain where the levels of TRK signaling may dip at times, that can mean for that cell, oh, I gotta die now.